multiple dominant alleles for a gene. If you've noticed that, like all the examples we've talked about, they typically have one dominant allele and one recessive allele, or maybe multiple recessive alleles, but always just one dominant allele. In this case, there are multiple. The major example that you will see with this is blood type. So blood type is a trait that has multiple alleles for the gene that controls it. Okay? And the gene for blood type encodes instructions for an enzyme whose job it is to catalyze a reaction that adds either carbohydrate, carb A, type A, carbohydrate B, or neither. This enzyme, based on the allele that's inherited, adds carbohydrate A, B, or neither. Sorry, one more thing. To red blood cells, RBCs, red blood cells. Okay? All right. Now you've heard probably the blood types, major blood types, type A, AB, B, and O. We'll get to the positive and negative thing a little bit later. But those major categories, A, B, AB, and O, right? So if you look at a red blood cell, I should use red. So a red blood cell, we're going to draw 4 of them. 1, 2, 3, 4. So if we have 4 red blood cells, you're going to represent what these blood cells look like in the different blood types. So if this is type A, this is type B, this is type AB, and this is type O. If you're an individual that has an allele for type A blood, on your red blood cells, that enzyme is going to add a little carbohydrate on the membrane of that red blood cell. Okay. So I'll make a little reminder. So in green, that carbohydrate A was added to the red blood cell, and so you have type A blood. For B, it's going to add a different carbohydrate. And type O, it's not going to add either. Now here's where the codominance comes into play. If you inherit the allele for type A and the allele for type B, neither one of those is recessive to the other. You actually get both carbohydrates on your red blood cells. So that's what's meant by this idea of codominance, is that both alleles are expressed in the individual. There's not a recessive or dominant allele. Okay? So, what you will see is that the allele, let's use this. So the allele for the enzyme that adds carbohydrate A, they actually use this little letter I with the subscript A. This is the allele for type A. This is the allele for type B. And this is the allele for type O. All right, so if you have, for example, an AV blood type, your genotype is IAIB. If you have type O, you are II, Captain. If you are type B, you've got IB and then I. And if you're A, IA, I. Okay. All right. Now the reason this is significant is because your immune system, its job is to recognize those carbohydrates. Do you remember when we talked right before break, we talked about juxtacrine signaling and how the immune cells will like get really close to a cell and sense if it's from that person or something foreign? It's how blood types work. The immune system recognizes these carbohydrates depending on what their blood type is, right? So if you're a type A blood type person and your immune system cell recognizes a B carbohydrate somewhere, it's going to freak out and start inflammation and cause some issues. That's why blood type transfusions, one of the major elements of that is determining which carbohydrates this person has on their blood cells. You can't mix those. Now, can someone tell me why type O is actually, you might have heard, like a universal donor? Why might that be the case? Yeah.
Type O were accepted by type A and type B.
Right. Why? They like fly. Yeah.
Is it because it doesn't have the carbs?
Right. So they kind of like fly under the radar. They don't have any like markers on them, but they're still blood cells, so they can do their job. But there's no identifiable marker, so they kind of just like fly through undetected. Right. So you've probably heard, right? There's also like AB positive, A negative, A positive. Just make a note to yourself. This is the positive, negative element of blood type is controlled by a different gene called the Rh factor gene, or the Rhesus factor gene. And this right here, this follows normal inheritance, where positive is dominant and negative is recessive. So the whole Rh factor, it's normal Mendelian inheritance, so you combine these two genotypes to get your blood type. Okay. We doing okay? Yeah? So codominance is our second example. While we're on the topic of dominance, the third example is incomplete dominance. Incomplete dominance. And the idea here is that the heterozygote is an intermediate between the dominant and recessive phenotypes. right? And the classic example here is that if you cross two of these true breeding flowers, they're called, major example here, are these snapdragon flowers. And if you cross two true breeding snapdragons that are either red or white, the F1 generation is going to be 100% heterozygous, and their color is actually going to be pink, not red. So they're like an intermediate of the dominant and the recessive phenotype, the dominant being red and the recessive being white. If you cross these again, if you do a monohybrid cross here, you're going to get red, pink, and white flowers. So instead of having a 3-to-1 phenotype ratio, you actually have 1 to 2 to 1 for both your genotype and your phenotype ratio. 1 to 2 to 1. Okay. That's incomplete dominance. It's just the idea that the dominant, in this case the red allele, doesn't completely mask the recessive, which is the white.
Mixed together.
Yeah, they mix together. Although, we got to be careful with the term mix here, right? Because one of the things that Mendel disproved was blending. Right? This is not blending. This is both alleles are actually being expressed. In this case, it's a pigment. There's actually not enough pigment being produced by the one red allele, and so you get this like lighter red or pink. Okay. So that's number 3. So the last 2. No, sorry, last 3. These are shorter, much shorter. Okay. The last 3 we're going to do right here at the bottom. So, number 4, these are polygenic traits. Polygenic traits, they refer to these as being quantitative traits. Examples like hair color, eye color, skin color, height. Right? Because if you think about it, there's not like just 2 types of hair color, for example. You have more of a continuum. Right? There's a quantitative nature to it that you can have either lighter or darker and everywhere in between for color and for height as well. So the reason that is because there are multiple genes involved. Typically, there's like between 3 and 6 different genes involved for maybe, say, hair color. And so your genotype for hair color isn't like big B, little b. It's a string of like 3 or 4 genotypes. Right? So those are polygenic traits. Okay. 5th one. Non-nuclear inheritance. And Mr. Anderson talked about this. I'm not going to go into too much depth here, but the idea is that, remember when we talked about endosymbiosis Chloroplast mitochondria, they have DNA in them. That DNA contains alleles, but mitochondria and chloroplasts DNA is inherited maternally. And that's because the egg or the ovum in plants, remember what we talked about on that poggle? Remember like the whole polar body thing and how they're destroyed, but the oocyte that matures is like the bigger one? It's because all of those mitochondria are being piled into that egg, but the sperm doesn't contribute any mitochondria, just the DNA. And so we have maternal inheritance. That's non-nuclear inheritance. And then the last one this is called phenotypic plasticity. And the idea here, plasticity means like malleable, or it's able to change. It's not stagnant or completely determined. And what this means is that you can think about this in terms of like environmental impact. Because there are traits like height, skin color, intelligence, and even, for using a plant, leaf size, that can all be influenced by the environment. So, for example, height has been shown to be influenced by nutrition. There are polygenic genotypes that can affect height, but actually nutrition, what someone eats, can influence their height phenotype. It's also been shown skin color can be affected by UV exposure. Right? So you inherit certain alleles for skin color, however, UV exposure can affect the phenotype. All right. Intelligence can be affected by experience. And leaf size can be affected by sun exposure, soil contents. Right? So the main thing here is that there are some traits that, although you inherit certain genes that affect the phenotype, that they can be heavily influenced by the environment. And there's this kind of dual play of environment and genetics for a lot of traits. If you took my health class back when I taught health, did anybody take my health class? A couple of you. One of the topics that we talk in health class is about mental illness. And mental illness, for example, like depression, major depressive disorder, or anxiety disorder, that there is research being done about genetic predispositions, but there is a lot of research as well about how environment plays into that as well. There's kind of this dual play of environment and genetics that can make an individual more or less likely to develop a mental health disorder. So like, for example, trauma. Like trauma is an environmental impact that can influence someone's mental health, right? But there are actually predispositions for major depressive disorder. Okay. So, I'm going to pause. There's one more that I'm like a little nervous to go into because it's a confusing one. So I'm going to give you like a break for about a minute, and then we're going to go into that last one together. Okay.